aqui no nosso evento. Então, eu já quero começar agradecendo a presença de vocês. É muito importante que vocês venham prestigiar os eventos que a gente promove, dá bastante trabalho, a gente tem uma equipe gigante por trás de todo esse preparo, né? Então, eu gostaria de agradecer mais uma vez a toda a equipe da LAS, que está promovendo esse evento, né? Também a PROESC da Universidade Federal de Pernambuco, a Unipampa do Rio Grande do Sul, né? Por todo o apoio ao nosso evento. E também a Pixel, que está patrocinando o terceiro SIMPECE. Só alguns recados para vocês, né? Então, a gente está tendo aí os minicursos, amanhã a gente ainda tem alguns minicursos, então não esqueçam aí do horário do minicurso de vocês, estejam lá no laboratório, qualquer dúvida vocês podem contactar aqui a equipe da LAS, tá? É bem importante também que vocês se atentem à confirmação de presença no evento, porque isso vai garantir o certificado de vocês. Tá? Então, ontem nós já liberamos o formulário ao final do evento com a palavra-chave e hoje também vamos fazer o mesmo. Tá? Então, ao final do evento, vocês vão receber uma palavra-chave e um link no Forms para vocês preencherem essa palavra-chave. Então, fiquem aí ligadinhos, tá bom? E também fiquem ligados lá no nosso grupo do WhatsApp, onde a gente tem sorteios sempre de brindes, né, e presentes para vocês, tá certo? Então, hoje, nessa noite, a gente vai ter uma dinâmica um pouquinho diferente, né? Nós temos palestrantes de fora do Brasil, e por isso também a nossa apresentação ela vai ser em inglês, certo? As palestras elas vão ser traduzidas, então vocês não precisam se preocupar, a gente vai ter legenda automática, mas é bom porque a gente já pratica um pouquinho com vocês também, né? E vocês também assistindo eventos com palestrantes internacionais. Eu queria pedir também para que vocês deixassem as perguntas de vocês no chat, né? Não fiquem tímidos aí, deixem as perguntas de vocês. Eu tenho certeza que muitos aqui estão curiosos sobre as pesquisas desses professores, né? É uma grande oportunidade também de network para vocês. Então, aproveitem esse momento para vocês conhecerem os professores e fazerem as perguntas que vocês desejam interagir aí no nosso chat, tá bem? E agora... acabou o português aqui no SIMPECE, a gente vai começar a falar inglês. So, welcome everybody, everyone who came from other countries to participate of our event. And tonight I will have a special guest here to help me to present this event, this meeting, the second night. And I would like to introduce you, Erika. Hello, Erika.
Hi, Priscilla. Good evening, everyone. It's a pleasure to be here today.
And Erika is a PhD student from Federal University of São Paulo, and she is actually now in the Burke Institute, which is the same institute of our first speaker of this night. So thank you so much for helping us today, Erika. And I will let you introduce and invite our first speaker.
Okay, so thank you, Priscilla, for the invitation, and thank you for the kind introduction. So good evening, everyone. Again, I would like to thank you the presence for all of you to join us today. So today we're going to begin our international session, as Priscilla told us. So we have like many speakers from different places. So we're going to start the session, and I'm honored to have Dr. Gordon Lithgow from the Buck Institute. So a little bit of his background. He grew up in Scotland, where he received his PhD in genetics. And following that, a brief postdoc in biotechnology in Switzerland, he joined the Tom Johnson's lab at the University of Colorado in the USA. And during his time there, among many high-impact publications, one of his exciting discoveries was elucidating the link between the heat shock and the heat shock response and the longevity using the C. elegans model. So then he started his own lab in England, and after a collaboration with another professor, Dr. Simon Meloff, a member of the Buck Institute faculty, they published a great groundbreaking paper detailing the first use of a drug-like molecule to extend the lifespan of a living animal using the C. elegans model. So after that, Dr. Lithgow decided to become affiliated to the Buck Institute in California, where now he is a professor and vice president for the academic affairs. Dr. Lithgow is a pioneer in the aging field, and he is recognized by coming with the term geroscience, a concept that now it's used worldwide to describe the research focus on the connection between aging and chronic disease. So right now, Dr. Lithgow's lab focus on innovating projects employ the C. elegans model for drug screening and to identify compounds that can extend lifespan and health span to gain some insights into the factors driving health span and the origins of the age-related diseases such as Alzheimer and Parkinson's disease. So thank you so much, Dr. Gordon Lithgow, for accepting our invitation. It is an honor to have you here.
Thank you very much, Erica. What a nice introduction. Good evening, everyone. This is great to be amongst C. elegans friends. It's really a very nice conference and international and different subjects. Fantastic. Yeah, as was said by Priscilla, Erica's at the Buck right now, and that's what the building you can see in the photo. It's a freestanding institute, non-profit, devoted to aging research, from the biology of aging all the way through to age-related disease. So, if you're in Northern California, please come and visit. It's a beautiful building.
It is indeed.
I'll start with this disclaimer. Myself and Julie K. Anderson are founders of a company called Symbiont, and there's data that I'm showing today really led to the formation of that company. Okay, so I'll get to worms because that's my favorite bit. But before we get there, I want to talk about the sort of current state of aging research and as a whole. And then what I hope to do is convince you that the worm has an absolutely key role in understanding chronic disease. So here's this worldview that's not accepted by everyone at all. Most people haven't even heard of it, most clinicians. But really it gets down to this idea that aging is the cause of chronic diseases in late life. And you look at this chart and you think, how is that possible? You know, I've got Parkinson's, Alzheimer's, cancer, osteoarthritis, you know, cardiovascular disease, sarcopenia, very, very different diseases. And it's hard to really get your head around the fact that there's something about aging is actually a cause, a trigger for all of these diseases downstream. But that's the idea we're really testing. What comes out of it is that if you target aging by whatever means, small molecules or any ways you can think of targeting the aging process itself, then the idea is that you would affect not one but more than a single disease. And the research that sort of leads up to this we named geroscience, and that Erica introduced this word, and it really is the interface between the biology of aging and age-related disease and making connections between them. Okay, so this is a view, it's a hypothesis, if you like, and it's not accepted by most people. So here we go. Can we apply the geroscience idea? Yes?
Just a second, sorry to interrupt you, but can you click on hide on your screen? Can you see the subtitles?
Okay.
Oh, perfect. Thank you.
Yeah. Sorry. Can I move this little box as well? Yes, good. Right there. Okay. So the idea is that we apply the geroscience concept that aging, that the disease is caused by aging, and therefore target aging. And then, you think about this, you think, well, surely this has been done by, big corporations who must have thrown tons of money at this. And that's not true. Companies tend to study diseases in isolation. Of course they do, because that's how they get patents and that's how they get approval for drugs for single indication areas. So this idea has not really been tested in big pharma. It is being tested in smaller biotech companies. And if anyone wants to talk about that later, That's great. But what's the pipeline? If you think about the pipeline in industry, it goes from millions of small molecules, drug-like molecules, filtered down through various screens into a few hits that go forward to treat whatever disease you're hoping to do clinical trials in. And so now we're thinking about, well, is there an aging intervention pipeline? No, and that's because not enough people are really doing this kind of research. But what I want to show you is that if we did have a pipeline, I think it'd be very effective in generating really good quality interventions that we could take into clinical trials. And the worm has a key role to play in all of this. Okay. Trying to advance. It's not working. There we go. Okay, so how do you target aging? Well, here's a controversial comment. If you're not making animals live longer, you might not be targeting aging. Now, you might be studying the fibrosis, and you might be studying the downstream effects of that, or you may be studying causal factors. Are you studying aging? Possibly, if you're looking at age samples and you're looking at human age samples, that there's no other way to do it really, and therefore, you might be looking at aging. But if you think about if you're making an animal live longer, what you're really doing is that you're improving the capacity, the function of so many different components of the animal. And I think making an animal live longer is quite amazing. We didn't know if it was possible. And it might tell us something about aging as a whole. And I think Erica mentioned Tom Johnson. And this statement that I just showed you really comes from Tom's thinking. He said that there's 100 ways you can make an animal sick, but there may be only one way that you can make it more healthy than it normally is. And he was the first person to show that you could indeed do this with a mutation in a single gene, which he named H1. This is the paper back in 1987. And the title, the first part of the title is the main part, mutation in H1 lengthens life. Second part of the title actually didn't pan out. Okay, so what did we learn from Tom's work and those that followed? Well, first it was easy to modulate aging. Who knew? And also increase health span. That's something we can maybe talk about later. Well, we will actually talk about it. To most people, the effects that Tom saw and others after him were surprisingly large. If you got 20,000 genes and you're altering one and you see a lifespan extension, that's kind of mysterious. You see these large effects. Similar genes were driving aging in different species. They started with worms and then flies and yeast. And it's complex. It's a wide variety of biochemical and physiological processes are impacted by the mutations that extend lifespan. So this isn't a clock. This isn't a single component that's driving the aging process here, in the sense that it's complex, that things are changing all over the place. Okay, very quickly, the worm. All this. This is really the genetics of longevity. And these are the different categories of things that have been studied in terms of longevity in the last 20 years. And basically, you can pick your favorite bit of biology and study it in an aging context in the worm. So mutations in these pathways or overexpression of genes in these pathways lead to changes in lifespan. So we know we've got a rich collection of targets to actually go after these with small molecules. Okay, so just a quick description of a pipeline and what we've been doing at the back for many years now, and that is we start with as many small molecules, drug-like molecules as we can. These can be synthetic. Many companies sell drug-like molecules, and they are man-made. They could be nutrients. They could be endogenous metabolites. So we've looked at natural product libraries as well. And we've moved those into worms and human mouse cell culture for the most part. And then on the rare occasion, we move them into mice. And I'll talk about an example or two of that. So I think the worm is really a platform for longevity therapeutic discoveries. And again, that's a big call, but we have some evidence that this could be the case. So very briefly, you know all this, but we do our survival assays in a pretty standard way, very similar to the way we do any experiments in C. elegans, using agar plates with a bacterial food source. We make the plates, we then add the drugs. They're not as beautiful as this one here. And then a day later, I should have had that on there, 24 hours later, we then start our survival analysis. And this is a normal classic survival curve you see here. So simple, you're coming into the lab, you're checking if they're alive or dead over the course of a month. And here the controls are starting to die at day 12. Maximum survival is like 27 or something. And then this particular drug is pushing the curves to the right. Okay, that's what we're looking for. Now, the aging field has candidates for clinical trials right now, and there's big efforts to get some of those going, particularly metformin. What you're looking at here is maybe the top 5 or so drugs or nutrients that are being studied for clinical trials. And you'll see the number of publications associated with rapamycin and aging at the top there, 2,600. So there's a lot going on. And even if you cut that down to lifespan itself, what's rapamycin doing to lifespan? There's something like 1,100 publications. So that's great. And rapamycin is already a drug. It has some drawbacks, but there is a promising way to go. Nicotinamide metabolism has thrown up all sorts of things, NAD+. Again, a large number of papers on aging and a few on lifespan. Metformin is actually the star right now, the superstar, in the sense that money is being raised for a large clinical trial that is approved by the NIH. Again, lots of papers. Vitamin D, lots of papers. Quercetin, lots of papers. The problem is, you get to the end of this list, and I could list another 5 or 6 or so that are promising, but really haven't been studied as much as these. And that's a problem. If we're talking about a pipeline for interventions, this is not necessarily the way that you would go. I mean, most of these were based on guesses as to what would happen. And actually, you can see that for the majority of them, the lifespan effect was first seen in C. elegans, but they didn't originate from chemical screens as such. Okay, so big challenges in bringing this about. I won't go over them all. If you even just go down to the bottom 2, there's all sorts of gender-specific effects we know about chemical interventions in mice. We're not capturing that in the worm unless we really go after male populations. We do know that genetic background has a huge effect on drugs. I'll touch on that, I think. We're part of a consortium of 3 labs, Patrick Phillips and Monica Driscoll, who are studying the variation between labs and between replicate experiments with combining different genetic backgrounds in wild strains and drugs. And it's extremely complicated, and that's a different topic. So challenges are there. If you go to the right-hand column here, you'll see the kind of things that we've been screening for. And on the left-hand column, you see the screens themselves. So we screen on lifespan. And we go into details about how to do that, but we can start with tens of thousands of compounds, do a lifespan analysis and cut that down to 20 or so that we want to work with. Lifespan surrogates are handy. I'm going to show you one. So it's something that we really believe is connected to lifespan. And you can also go after race-related disease pathologies, and these are strains that have been created, usually expressing human proteins that mimic certain aspects of human disease. Okay. Lifespan surrogate, we're going back to what we did in 2006, where we published this paper showing that checkpoint proteins were involved in lifespan. We did this by selecting for thermotolerance, thermal stress resistance in a genetic screen. And those worms that were resistant to heat shock, many of them were also long-lived. So this was a genetic screen, and now we're basically doing this as a chemical screen. Not to go into all the details, but this is done quite differently from the lifespans, doing them in microtite plates, and we're agitating those plates. Get back on the screen here, and we're agitating those plates for 24 hours, and then we slam them with a 35 degrees or higher heat shock and score them manually. But we're using cytox, which is a dye that dead worms take up. Those complications with drugs in here, but for the start, the acids were quite well. And
Pardon?
This example is
excuse me, Gordon. Sorry to interrupt you again. Can you hear me?
Yes.
Gordon?
Yeah. It's very scratchy.
Your connection, it's a little bit poor. It's bad. So can you pause just a little bit to see if can stabilize or something? Can you hear me okay?
Yes, I can.
Okay. So guys, just it seems that we have like a little bit problem with connection. So he gonna check.
Let me yeah, I'm gonna try a different power pack. Hold on, please.
Okay.
Okay. Just wait.
Okay. I can hear you, but the connection is a little bit like poor. Like I can not hear properly.
Okay, how's that now?
Oh, now it's perfect. Okay, I can hear you perfectly. So sorry about that. To interrupt you. Okay, go ahead.
No, it's my fault. It's my fault. I had a power pack that wasn't very good. Should I go back a slide, or are you okay?
Yes, I'm okay. You can go ahead.
Okay, so I'm talking about some hits that have come out of the thermotolerance screen, and these compounds are metabolites of methylene blue. Methylene blue has a huge literature, by the way. Some of these compounds do as well. You can see some things from the literature on the left here. Proteasome activity, for example, is very interesting to us. Anyway, the reason we're excited about these compounds, here's Azure A, is the magnitude of the lifespan extension. This is rare. Usually we're around 35%. I think this graph's at 50%, and we've seen up to 80% with some of the experiments. So this is great because a large difference in lifespan really gives you something to work on. The problem is, when you ask, well, why such a large lifespan extension, and you think about, well, what's known about these compounds that would explain it? The answer is nothing. We've no idea. And we're working hard on these because we are excited, and hopefully they'll end up in mice at some point, but we do need to do a lot of work before then to work out what's going on. So instead of talking about the recent hits, I'm going to talk about hits that are published, but I've organized this hopefully in a way that kind of makes it interesting the way these compounds work and the fact that they might all work the same. Okay, so we've got a synthetic compound, thioflavin T, binds to amyloids. We've got alpha-ketoglutarate, metabolite, TCA cycle, does lots of other things as well. And then we've got vitamin D, which needs no introduction, a nutrient. And here's the published data from our lab and the Hong lab on EKG, alpha-ketoglutarate. And we don't need to go through them all. We'll just go to the first one, thioflavin T. The control curve is in black. And once you add thioflavin T, you can get up to 70% increase in lifespan. Interesting compound. We published in a prominent journal, mainly because this compound is already used in humans to visualize amyloids. So it was quite interesting to see a biological effect. We all thought it was inert before then. Okay, so different compounds, all extending lifespan, mechanism of action. So I'm taking you to a series of models, and in this case, mainly the A-beta model. So what's going on here is that Chris Link and many others since then are expressing human A-beta, turns out to be 242, not 142 in this particular construct, but they're expressing A-beta in C. elegans. And this is the midriff of C. elegans, and the white arrows are pointing to bodies, puncta in the muscle. So these form over the course of like 10 days. And we think of these really as amyloid deposits, a bit like what happens in the human brain. So this happens, and many of you know that when you do this, the worms become paralyzed. And that's an amazingly lucky tool to be able to use. Oh, 342, sorry, not 242. Okay, so this is a control animal. It's accumulating these amyloids, and it has a paralysis phenotype. Thioflavin T, you can see the signal here. It's immunohistochemical stain, by the way. You can see the signal here of the soluble A-beta, but there's no puncta. And this animal is not paralyzed. It's crawling around on the plate just fine until we grabbed it. So interesting. Thioflavin T is able to suppress this problem with proteostasis. Let's keep it that way. And it turns out that despite the very different structures and the very different origins of the other compounds, AKG and vitamin D, they all have this effect. So Thio-T, so this is paralysis now we're looking at, percentage paralysis. These are different experiments on each dot here. And you can see very clearly that thioflavin T suppresses this paralysis quite dramatically. AKG, not as dramatic, but also suppresses paralysis. This is graphed in a different way, but you're comparing the black to the white lines, and there's clearly also suppression of paralysis with vitamin D. So that's interesting, I think, at least, that three compounds that are very, very different are all having this effect on paralysis. And actually, when we look at more compounds that came out of a different screen, this is a graph constructed by Grace Jones, University of Oregon, using our data. And if you're below the line here, you're less paralyzed. And these are different compounds at different concentrations. And you can see that basically almost a third of the compounds we're testing here are preventing paralysis. So this is amazing, I think, that you go after lifespan. You find compounds from screens or from good guesses that affect lifespan. And lo and behold, you've enriched for compounds that also are able to prevent A-beta toxicity and aggregation. That's way beyond a coincidence, and it's an incredibly high hit rate compared to any other chemical screen you can think of. Okay, I lied about Azures. We do know at least one thing, and that's that they do the same thing as well. This is very preliminary data, but seems like a clear effect of adding at least 100 micromolar. That should say Azure there, Azure A. So interesting. Okay, so we continue to work on these things in the worm, but I'm going to very briefly, and I'll skip some slides since we're probably behind the clock. But I want to talk about how these compounds perform in mice. So thioflavin T, interesting design here where you're following individual mice longitudinally. So you're looking at the same mice again and again and again. This is a design by Simon Melov at the back, but lots of investigators involved in this. So you bring in old mice, you do a drug intervention, and you measure different things at different times. And the big superstar result from this was that thioflavin T, or at least a compound very like thioflavin T, is able to reduce bone loss in mice. So mice lose bone just like humans do, osteoporosis, and the thioflavin T-like compound delays this by 30%. AKG, TCA cycle, does lots of things because it's a cofactor for various enzymes. We did a similar study design starting late in life and doing all the measurements throughout. And in this case, we couldn't do the fancy stuff that we did in the previous study. We just didn't have the resources. But what we did do is lifespan, and we did 2 cohorts. We thought that was important because the effect sizes you see here are actually quite small. So these are pooled female and male. The males actually do better alone than the females. And you can see in both cohorts, there's significance, at least something like 16%, I think in cohort 2. We then moved to a mouse frailty index that designed by Susan Howlett. And the frailty index is basically a measure of everything you can look and see, kind of like holding a mouse in your hand. What can you do to measure aging that isn't going to involve complex instrumentation? I'm not going to go into all the details, but these are relatively easy assays of function and frailty. What you're looking at is a series of panels that where things are going quite well. Confine your attention to top left. The control animals are going up in an index. Here happens to be fur color. So the controls are going from black to gray, as measured by what seems to be quite a subjective measure. But actually in aggregate, I think it captures what we're looking for. And the AKG fed mice are on the bottom. You can see they're just bouncing around here. There's really no increase at all in the fur coat color. And although the differences are smaller, you can see there's trends for many of these assays separating, especially at later months. And again, these are the same animals being tested over and over again. Quick picture, mice on the right. You can see the gray coat. Also, if this was a video, you'd see they're not walking very well. Generally behaving like aged mice. The mice on the left are the AKG treated. The coats are black. They're moving normally, and they just seem like younger mice. Our interpretation of this is a little bit complex, but I'll mention it, and then if we've got a chance to take questions, we can do that. And I'll try and finish as soon as possible. Our interpretation of the measures that we made is that we actually compressed morbidity in late life. So in the controls, you see the pink triangle represents illness in late life, starting at the 75th percentile of death. And then the alpha-ketoglutarate, you're pushing out the health span to 95% of its lifespan, and you see that you shrink the morbidity. So you're extending lifespan, you're extending health span, but also importantly, you're compressing the period of illness. That's amazing to me. I have no explanation for that whatsoever. No idea what's going on. So is the worm a good idea in the end? Yes, and actually 40% of the longevity-inducing compounds have beneficial effects in preclinical mouse studies. This is our observations, published and non-published. If you took the literature as a whole, it wouldn't be as good as that. It'd be much less, and that, I think that's because of the way that people choose compounds for mice. Anyway, we do think it's a very effective way to find compounds that are useful in mice. Very quickly, we're continuing to do this, and I just wanted to mention this paper that was published 2 weeks ago now. And this is a paper from Julie Anderson's lab at the BUP in collaboration with myself, Manish Chamali's the lead author, and actually, so myself, Malene Hansen, and others were involved in this paper. And it's a discovery of a molecule that engages a nuclear hormone receptor pathway, FXR, for its effect. And it not only affects lifespan, but it also affects mitophagy as well. I think I'll just skip the screen here and go to the general things that we've seen with this compound. The top stuff is the stuff that's in the Nature Aging paper, showing lifespan extension and dependency on FXR-like pathway. All done in a worm, by the way, then validated in cell culture. So the worm is leading the way through most of these things. We've gone on, this is unpublished data, to find a direct target and examine drug-like properties, and hopefully PD and AD models are initiated and will be interesting. Okay, so just another example going from through the worm, actually. This example shows you start in human cells. You do a screening to human cells. You move over to the worm to get mechanism, and then back into cells to corroborate that mechanism. So it's kind of an interesting way in the structure. So what's next? Mechanism. We need to understand how these things work. New compounds, of course, different models of mouse aging and disease, and hopefully clinical trials. I want to mention this paper now. I'm closing here. It's a paper from Vijay Yadav's lab at Columbia, and it's not our paper at all. It's Vijay's. There was something like 50 authors on this, but 7 of the authors are based at the BUC. And those authors based at the BUC provided through C. elegans evidence of a lifespan extension that you know, there was no other way to do it. This is an extremely comprehensive paper with data from humans, primates, rats, mice, worms, maybe flies as well. And it's the kind of paper that I think shows the way in which the worm will always contribute to aging papers, no matter how preclinical they are. And that is to provide the evidence of lifespan itself, going back to the idea, if you're not making an animal live longer, you might not be working on aging. Great paper from Vijay's lab, and well worth a look, just to stand back and go, wow. I think I finished there. Yes, I do. Just thanks to members of the lab. Many of the names here were involved in screens. Shankar Chinta in Julie's lab. Manish Chamalai did all the work on the mitophagy compound you saw at the end there. And most of the mouse studies were done in collaboration with Simon Melov and with Brian Kennedy. Thank you very much.
Amazing talk. Thank you so much, Gordon, for being here today. So we have a few minutes for questions from the audience, but before that, I wanted to ask you something. Actually, this aging field, like, it is absolutely one of my favorite topics, but I wanted to know, like, in your opinion, what it means health span, because I know that each lab has like their own definition. So I wanted to know, in your opinion, which is a good like parameter or a good definition for health span in C. elegans.
Oh, good. You know, I used to think this was easy. If an animal was crawling around a plate, it must be healthier. But what we see is these mutations in things like insulin signaling, where the animals are living a long time, but they're not moving around the plate. They're just sitting there. And it's widely recognized, I think. And there's some debate about how to analyze this, but I think it's widely recognized that there are examples of mutations where the health span is increased. And how do you measure it? Mainly movement, to be honest. And movement is a good way to capture both muscle and neuronal activity. You take out any one of those and you're not moving. So it aggregates at least two different tissue types and their function. So mainly movement. You can do this in an automated way. There's lots of systems to do it. It's difficult, because even movement itself has all sorts of features. And do you choose body bends? Do you choose distance covered? There's lots of things that can be measured. So my problem started with health span when I was talking to a physician, and he said, this health span thing, and I'm going, yeah, I think it, we know enough to sort of measure it in worms. And he said, well, how about this? He said, patient comes in, high blood pressure, we give them statins, they go away, they're normal. Is that a healthy person or an unhealthy person? And I went, oh, never thought about it like that.
No, yes, definitely. So we have another question from Lucy Clade. Actually, you kind of explained a little bit when you mentioned the paper from Julie and Manish, but Lucy Clade is asking, how does the study of the longevity of C. elegans related to understanding the longevity in humans?
Yes. Well, this goes way back to when the first genes were cloned in the aging pathways. And it turned out, they weren't necessarily aging pathways. The age one that Tom Johnson isolated was a PI3 kinase. The next gene that Cynthia Kenyon identified, and then I think she cloned it. Yeah, she did. Turned out to be involved in insulin signaling. So very quickly, actually, all the proteins in insulin signaling pathway were tested for lifespan, and many of them are recorded as having lifespan increases. So insulin signaling, it's just a direct connection to humans. The gene, the proteins are the same, and that goes for almost all those pathways I put up that have been identified, but they have human homologs of those pathways. There isn't really a pathway where we're going, humans don't have that. That's interesting. There's very few genes. I'm sure there are. There must be worm-specific genes for lifespan, but most of them have human homologs. So then the epidemiologist, the genetic epidemiologist, showed that one of the genes in worms, FOXO, also came up as a hit. Very rare, but it came up as a hit for centenarians. In other words, having a certain variant of FOXO, which was shown to, important in lifespan extension, also showed up as a gene that affected longevity in humans. So since then, it's not. I guess it's not being acted on very directly, other than the potential clinical trials for small molecules. I think that will be the ultimate evidence. When we have 5, 6, 10, 15, 20, 100 compounds in clinical trials that affect aging, we'll see what the success rates are like, and that will tell us a lot about the similarities between worms and humans.
Okay, thank you. We have another question from Professor Diana. She says, great talk, fantastic work. He asking, we were trying to validate the Fradin index of C. elegans using different endpoints. We expose worms to airborne toluene and observe the behavior, mitochondrial function, neuronal viability, lipofuscin levels, for example, as an endpoint to compose the Fradin index. Which other endpoints would you suggest? We found that for mammals, there are approximately 70 endpoints. So do you have any suggestions for that?
That's really a great point. And well done for taking that on and generating the kind of data you're generating, because we definitely could do with other surrogates And as you say, like a panel, a frailty index for worms would be fantastic. Nothing comes to my head immediately. Obviously, the morphology of the worm changes with age. Not necessarily an easy assay, but for example, very old worms cell dropout in the intestine. And in fact, you see breaks in the intestine, it just disappears. I don't know when that starts. It's a dappy stain, so in some ways it's relatively easy, whether you could screen on it, I don't know. But I think as a health span indicator, you know, losing parts of the gut is obviously a dysfunction. So that's something. And then Kathy Wolko many years ago published a series, well, not a series, I guess one important paper showing changes in the morphology of the pharynx with age. And she used an AI tool to look at the images and come up with, you know, an image that represented a sort of health span drop off or, you know, a change. Actually, I think she identified 3 phases of changes in the pharynx. And, you know, unfortunately, I don't think that work's really been followed up on in the way that Kathy was doing it. So yeah, I'll try and think of more. My lab actually talks about frailty index quite a bit, so they're thinking of things that they could measure.
Okay. We have time for more, just 2 questions. So this one is for Esther Lopez. She's asking, what is the correlation between restricting caloric and the longevity?
Well, it's pretty good. You probably know that in mice, it works on the lab strains, but there are many mice strains that it doesn't seem to work on. Now, it's controversial, but there's 2 labs took a panel of inbred strains, recombinant inbred strains, they're all genetically different, and they put them on a single regime of diet restriction. And as expected, you know, the mice from the lab did exactly what they always do, live, excuse me, my voice is not good today. They lived about 40% longer as usual. But then many of the recombinant inbred strains did not respond at all. And actually, in some, the caloric restriction actually reduced their lifespan. And you can argue about, well, each strain needs its own dose, if you like, its own level of intervention. I am sure that's true. I think if you actually were to do different doses, you could probably find for many of these strains a dose that would work. In worms, caloric restriction extends lifespan. That's the first thing that we check with new compounds is whether they're having an effect on caloric restriction. We do that by measuring the amount of food that they're taking in. We can compare them to caloric restriction mutants and do some genetic crosses and so on. So it's real in worms for sure, at least in the lab strain.
Okay, so we have like our last question. It's actually from our next speaker, Alex Benedetto. So he's asking, does alpha KG improve mouse cognition, like memory, executive function, etc.?
Yeah, we have not looked at that, and actually just over the last year and a half, we've sort of we have a lot of equipment for functional aging, muscle, heart, you know, metabolism, and so on, and we were kind of lacking sophisticated equipment to do the behavioral stuff, but we've got it now, and we'll you know, if we can get the resources, we'll definitely do it. I think that'd be super interesting.
Okay. So thank you so much, Gordon, for making today. I know the schedule was a little bit tight because of the time zone. So, and I know it is a holiday week. And so in name of the organization of the SIMPECE, I want to just say thank you. We really appreciate your presence here today.
Thank all of you. Thanks for your questions. Thanks for your attention. That was really fun. And looking forward to the next talk.
Okay, great. Happy Thanksgiving and see you then.
Goodbye.
Bye. So everyone, this was our first speaker. I hope you guys enjoyed the speaker. And right now I'm going to introduce our next speaker. Just a second. Now I'm very excited to announce Dr. Alexandre Benedetto. He's going to be talking about the whole of quinurenina pathway in C. elegans host gut bacterium. But first, let me introduce him, like a little bit of his background. So Dr. Benedetto, he is currently working as a lecturer in the Department of Biomedical and Life Science in Lancaster University at the UK. His research is interested to understand how animals and their microbiome communicate through life to maintain homeostasis. To probe the social interactions, he studies the C. elegans and its adult gut microbiota under challenging conditions such as infection, neurodegenerative disease, stress, and aging. Also combining genome-wide phenotype screens, he aims to map the genetic network that regulates warm gut bacteria interactions. In parallel, he also studies the regulation of abrupt death in C. elegans in order to identify interventions that can delay tissue decay and also organism death. So with that, I would like to thank you, Dr. Benedetto, for accepting our invitation.
Yeah, can you see me? All right. Yeah.
Hello. Okay.
Thank you for the invitation. It's going to be a tall order to follow after Gordon. It's kind of like a summit in the field, but luckily I'm working on something slightly parallel, so I guess the comparison will be okay. So yeah, that was a fantastic talk earlier. I don't think I think it would be difficult to find a better speaker to start this session. So should I start right now?
Yes. Feel free to start a presentation. Just a second, going to share your screen. And I just wanted to say that it is an honor to have you here today. So once again, thank you for making time for this. And I think we can see your screen now. So I think you can start it.
Okay, very good. All right, so good evening, everyone. Evening for most of you, I guess. So today I will talk to you about how the worm quinurenina pathway is implicated in the regulation of gut microbial content. So that's work that has been going on in my lab for the past 5-6 years mostly, and which is yet to be published. For those who have not been able to attend the recent C. elegans meetings, they might not have seen these results yet. So as you know, like everybody else, I guess, on this video call, or most of us, we're working with model organism C. elegans. And in my case, I'm particularly interested in how it interacts with gut microbiota, also gut pathogens as well, occasionally. They are actually very convenient to screen for interactions between host and microbes, because obviously the screenable phenotypes are really easy to deal with, which is usually survival. And so this is the sort of things we do. We transform microbes and then we look at what they do in C. elegans guts. But recently we've also been doing a little bit of work on imaging essentially brain activity at death, but also I will want to combine that with microbial exposure in order to get a higher, deeper understanding of the gut-brain axis in C. elegans. So the story I will talk to you about today stemmed from a work that I've done in actually the James lab started in 2012. And I was involved in a study where we reported that when worms die, they start fluorescing in blue. And the culprit for this, increase in blue fluorescence in the gut, so that's not the total blue fluorescence of the gut. There are other compounds that are blue fluorescing in the gut, but this is the fraction of the blue fluorescence that increases at death. And we found that actually it's due to the quinurenina pathway, which produce those. I'm going to get my pointer. Sorry, those compounds down there on this arm of the pathway that are blue fluorescing. So antranic acid itself and 3-hydroxyantranic acid are both fluorescing compounds, but the compounds that are majorly responsible for the increase in blue fluorescence in the worm gut when they die are actually esters of enteric acid. Okay, so the question that that we asked then was, fine, the worms are glowing in blue when they die, but for what purpose? And what are these blue fluorescent compounds doing in the gut granules? So looking at the quinurenina pathway, it's the catabolic pathway, the main catabolic pathway for tryptophan metabolism. It produces a large diversity of compound down there. You see enteric acid in here. And tryptophan is right there in the middle. And about 95% of the quinurenina pathway, of the tryptophan metabolism, actually goes through the quinurenina pathway in mammalian organisms. And perhaps one of the reasons for that is because of the quinurenina pathway, mammals produce nicotinamide, and the pathway can also be used to feed glycolysis. So it's quite important in terms of energy handling. C. elegans and other nematodes actually tend to not produce NAD from the quinurenina pathway, which is an interesting idiosyncrasy of C. elegans metabolism. But what are those blue compounds doing in the gut of C. elegans microbes, of C. elegans? So the quinurenina pathway in other organisms have been shown to be involved in metabolism, aging, host-symbiont interactions, even quorum sensing between bacteria, in animal development, but also neurological diseases, which is a big focus, and infection and immunity, which is another strong focus, especially gut immunity. In C. elegans, it was found years back now by the Nolan lab that the quinurenina pathway is indeed involved in aging as well, and in development, and notably also involved in energy metabolism in C. elegans. So no NAD production in C. elegans, but rhodoquinone metabolism is necessary for the worm and relies on the quinurenina pathway. And more recently, there have been two short articles from the Ashrafi lab, and that have demonstrated that the quinurenina pathway has roles in neurological diseases in C. elegans as well. But what role does it play in C. elegans gut physiology? So we set out to essentially try to understand that. So now if we look at the type of organelles in which the quinurenina pathway compounds are found in C. elegans, they're called lysosomal-related organelles or gut granules. And lysosomal-related organelles is a wide family of lysosome type of organelles that fulfill a wide range of functions in other systems. So, you know, melanosomes, for instance, are one type of lysosomal-related organelles. And then you have like dense core vesicles, and for instance, what is secreted also, the secretion of surfactant in the lung also involves lysosomal-related organelles. So they can have very diverse functions, but they are quite specialized compartments. And until recently, the function of C. elegans gut granules was quite mysterious. They're very prominent in the worm gut, but their exact function for the amount of gut granules that the worm has was not clearly elucidated. It was found that they were involved in zinc homeostasis, and very, very recently, actually, and I just discovered that a couple of weeks ago, it was reported that they also promote stress and immune responses in C. elegans, in particular in response to infection by Pseudomonas aeruginosa, which kind of confirms some of the findings that I will demonstrate now. So our hypothesis, coming on the back of this study on Blue Death, was that perhaps the gut granules in C. elegans and the quinurenina pathway compounds are actually involved in antibacterial defense. So we hypothesized that it was some sort of antimicrobial arsenal. And the reason for that was that in the previous literature, so back in 2011, we can see that in the context of gut infection and gut infection only, not other epithelial infection, allelobiogenesis genes are upregulated upon infection. And that is about gut infections from bacteria, but also from fungi. And on the other hand, we knew that quinurenine pathway metabolites actually are fairly bacteriostatic. Quite a few of them are bacteriostatic properties. And they're able to inhibit gram-positive and gram-negative human pathogen growth. So my idea was to take a pathogen that it doesn't have a specifically dedicated, very highly specific strategy to kill its host in C. elegans. And so I ended up choosing E. fecalis rather than P. aeruginosa that produces toxins. And E. fecalis is also highly relevant to hospital-acquired infections, as well as a commensal that can become pathogenic, which I found particularly interesting with my broader interest in studying the balance between microbes and worms. So the first question we asked was, do mutation in gut granule biogenesis or quinurenine pathway genes actually affect C. elegans resistance to infections? So we tested that classically, exposing different mutants of each pathway to pathogens. And what we found is that indeed some mutants of the lysosomal-mediated biogenesis pathway, in particular GLO1, which is the RAB32 homolog in C. elegans, are quite significantly more resistant to infection by E. fecalis than wild-type worms. And what we see in these mutants is actually that the blue fluorescence that's normally containing little blood granules in the gut is actually delivered in the lumen of the worms when they are infected with E. fecalis. So upon E. fecalis infection, there is an increase in blue fluorescence that's only found in the lumen. GLO1 are actually mutants called because of their glow phenotype, and in this case, because they glow less than wild-type worms, which means they have actually less blue fluorescence. And that's due to a defect in the formation of those gut granules. So this was very interesting because what it could potentially suggest is that GLO1 not having fully functional gut granules might be dumping essentially its normally gut granule content into the lumen of the intestine, and that would create an environment that is bacteriostatic and then basically slow down the infection process. So at the moment, we're still investigating that, but this fact was essentially arguing for a potential role of allelors in antimicrobial defense. So switching to the quinurenine pathway now, we did similar experiments. And so we exposed quinurenine mutants, the quinurenine pathway mutants to E. fecalis infections. And we saw that one mutant, very distinctively, the quinureninase mutant CANU1, previously called FLU2, was highly resistant to E. fecalis infection, to the point that they can actually get used to E. fecalis very well and propagate fine and feed off E. fecalis. And we saw that also in another type of assay than the plate assays. We also saw it in a liquid assay that kills the worms very quickly. And actually in that assay, we saw that another mutant, the KMO1 mutant, was significantly more sensitive to E. fecalis infection. And that will become a bit clearer later. However, now that we have mutants that can inhibit E. fecalis infection, we wondered how they achieve that. So we took kind of like more of, I would say, a chemistry approach here, very simple. We bought all the different compounds that we could commercially get from the pathway, and basically we exposed two different strains of E. fecalis to a range of concentration of these compounds. And eventually we found the ideal concentration for the treatment. And we made sure that the pH stayed stable at 6, because pH has a massive impact on bacterial growth. And in those conditions, we found that two compounds in particular were very bacteriostatic against E. fecalis. Those were 3-hydroxyquinurenine, and you can see in teal down there, and picolinic acid, which you can see in red here. And you can appreciate that different strains of E. fecalis do have slightly different behaviors. So next, we hypothesized that perhaps the way these compounds can be helpful to C. elegans when encountering E. fecalis is simply by being delivered in the gut, and there they would create a bacteriostatic environment, and E. fecalis will have more trouble to grow and invade the C. elegans intestine to trigger disease. So we used a fluorescent version of E. fecalis, a green fluorescent version of E. fecalis that was produced by Daniela Garcin. And we infected the worms with that, the different quinurenine pathway mutants, and we basically tracked over a period of 3 days how the gut fluorescence was progressing. So the gut fluorescence provided by the green bacteria. And what we found is that, unsurprisingly, the canu1 mutants were colonized both slower and also to a lesser extent, suggesting that one of the ways canu1 mutants managed to fend off E. fecalis was simply by reducing the gut load of E. fecalis pathogenic bacteria. So, next, we extracted, basically we made mushed worms. We took extracts of different worms of the quinurenine of the different mutants of the quinurenine pathway. And now what we did is that we exposed E. fecalis to these extracts to demonstrate that it is the extracts or the metabolites from the worms that are directly inhibiting E. fecalis growth. And what we found is that canu1 extracts actually very strongly inhibit E. fecalis growth. Actually, wild-type extracts compared to no extract already significantly inhibit E. fecalis growth, which was not necessarily something we would automatically have expected. So given that the 3-hydroxyquinurenine was previously found to be a very potent bacteriostatic in other studies, we suspected that that could be also the reason why quinurenase mutants actually are able to inhibit E. fecalis growth in their gut. Because in the paper from the Nolan lab, they reported that canu1 mutant contained really high level of 3-hydroxyquinurenine. And we confirmed that as well using a slightly different biochemistry approach, which was based on HPLC UV. So this seemed to indicate that canu1 mutant might be producing high level of 3-hydroxyquinurenine because of the quinurenase being mutated, and that has a constitutive effect of making them more resistant to E. fecalis infection. In itself, that doesn't make it a new immune pathway. In order to demonstrate that there is some sort of purposeful pathway, one needs to show that the quinurenine pathway activity can be modulated in response to a pathogen. So first of all, what we did is that we decided to look at what the autofluorescence of the worm looks like in the gut when we exposed the worms to E. fecalis. The reasoning here was if we modify the quinurenine pathway activity, we should get a fluorescence shift in the gut of the worm. And that is exactly what we got. So what we saw is that in wild type, after about 4 hours of infection, there was a significant rise in a specific ratio of fluorescence in the gut of the worm. And that rise was absent in the CANU1 mutant. So that gave us some confidence to try to go through slightly more expensive techniques. And so we switched to more omics sort of techniques that are a bit more involved and expensive. And so we did some RNA sequencing to try to look at the genes that are upregulated or downregulated upon worm infection by E. fecalis. And what we found by looking at the quinurenina pathway is that the KMO1 enzyme that sits right here was upregulated, but the quinureninase was downregulated. And that agrees with what we found earlier, because CANU1 mutant, which doesn't have proper quinureninase activity, is more resistant to E. fecalis. So we would expect, if our hypothesis is right, that the wild-type worm would try to downregulate CANU1 in order to fend off infections. We next moved to trying to measure those metabolites using HPLC UV. And what we saw is that upon infection, tryptophan is being converted more aggressively into quinurenina pathway compounds. And amongst those, there's a sharpest rise in the amount of 3-hydroxyquinurenine, as well as an increase in anthrenic acid. So our interpretation of this data is that upon infection by E. fecalis, KMO1 is upregulated, CANU1 is downregulated, and that leads to this increase in 3-hydroxyquinurenine that is able to inhibit E. fecalis. At this point, however, it appeared quite unlikely that this sort of mechanism, which involves such a massive, very, very central conserved pathway, would be specifically dedicated to fight a single sort of infection. And obviously, the very recent study that was just published and demonstrated that the quinurenina pathway, that the lysosomal-related organelles, are involved in defense against Pseudomonas aeruginosa, which is gram-negative, whereas E. fecalis is gram-positive, suggests that indeed those will not be responsible for very specific defense. And actually, we hypothesized that this might be a great way to regulate the gut microbiota composition, rather than simply dealing with gut pathogens. Another idea also that guided us in that direction was the fact that E. fecalis is not a C. elegans natural pathogen. So if C. elegans would have some sort of evolved pathway to cope with E. fecalis, it couldn't have done so by exposure with E. fecalis. So we went back and looked at published data sets on worms that have been shifted from one microbe to another. And we look at what kind of genes are getting up and down regulated. And when looking at the quinurenina pathway and its modulators on the top, we found that quite frequently several genes of the quinurenina pathway and their regulators are either up or down regulated when worms are shifted from one microbe onto another different microbial diet. And this is true in commensals and pathogens. So next, another indirect evidence that we got from the lab was that we were carrying out actually like wide stress assay, where like kind of quite intensively stress assay, heat stress, oxidative stress assay on worms that are exposed to different microbes to try to figure out if some of the commensal microbes of C. elegans can promote heat or oxidative stress resistance. And while doing that, we collected a lot of data on our plate reader, and we were measuring blue fluorescence. And blue fluorescence is an indirect readout for the amount of compounds on the anthrenic acid arm of the pathway that are present in the worm gut. So we plotted the amplitude of the peak of death fluorescence that we observed in the worms that have been grown on different microbes down here. And what we saw is that for the same amount of worm put in every single well, we had very different amplitudes of blue fluorescence peak. And that suggests that a number of these microbes here are actually downregulated, have either lower canarine pathway activity or downregulate specifically the anthranilic acid arm of the pathway, whereas others are upregulating it. So that gave us the confidence to go further and to invest a bit more resources to get to the bottom of this. And so we wanted to test now if the worm canarine pathway activity could impact natural gut microbiota commensal, and notably their ability to occupy and colonize the worm gut. So we performed a number of, again, similar to what we did with Ifecalis, a number of growth assays for all these microbes. And we found that they all behave in slightly different manners. So here you have some examples. So for some microbes, picolinic acid, for instance, is inhibiting growth quite strongly. Although, for instance, this microbe has a very high capacity to grow to really high densities. Other microbes will be mostly inhibited by anthranilic acid or 3-hydroxyanthanilic acid. And we actually found that L-quinurenina, in most cases, is actually acting almost like a prebiotic. We did expect this sort of activity from tryptophan, because tryptophan is often a rate-limiting amino acid, essential amino acid. But actually, tryptophan for some microbes, and here it's here in like darker purple here, and it can be guessed here as well. Sorry, it's covered by the other curve. Can actually be inhibitory as well for certain microbial growth. So to confirm this in vivo, we next decided to try to tag as many microbes as we possibly could in different colors so that we could use live fluorescence microscopy in order to track their progression in the worm gut as the worm ages. So typically what we would have, we make this type of plates where we have a couple of microbes together, 1, 2, or 3 microbes, and then we can feed the worms this, and then we'll get these sort of pictures, and we can try to assess the amounts of microbial colonization there is. At the moment, we try to move toward also characterizing the inside, I would say the organization of the microbes inside the gut of the worm by using super resolution microscopy approaches. But what we found is that looking at canarine pathway mutants and subjecting them to different microbes, KMO mutant and CANU1 mutants had differential impacts on very specific microbes. So here what you can see is that while other canarine pathway mutants and wild type enable a relatively slow colonization by E. coli, KMO1 mutants are colonized very, very quickly, and then the colonization of the gut is achieved early and remained roughly the same throughout the 5 days that the assay lasted. Conversely, in the CANU1 mutant background, one specific bacterium out of the 5 that we tested was inhibited, and that bacterium is Stenotrophomonas maltophilia, which is actually a really excellent colonizer of the C. elegans, the wild type C. elegans gut. And in the CANU1 background, its colonization was vastly diminished. Now, plotting data in a slightly different way, what we actually realized is that in the KMO1 context, it looks as if the worm is almost unable to normally discriminate between different microbes. Most microbes colonize at relatively similar rates when you compare to the situation in N2 control. And what it leads to is that KMO1 can actually make some microbes that normally are not pathogenic, like we see Ochrobacterium here, slightly pathogenic. by enabling gut colonization to an extent that is not normally seen in control worms. So with this, we thought we identified essentially a new modulatory pathway for gut microbial composition, and pinpointed one of the key enzymes involved in that, which is KMO1. So the current model that we're working with is that the balance between KNU1 and KMO1 activity regulates the composition in terms of quinurenine pathway metabolites, downstream metabolites, that the worm will be able to secrete in its gut lumen, and that will have an impact on the gut microbiota and its composition. So different cocktails, if you wish, of the quinurenine pathway metabolite will have a slightly different effect on different members of the gut microbiota. And the worm is able to regulate that, and in this way, it can ensure that its gut microbiota is in check. So we think we have uncovered essentially a major mechanism of regulation of the gut microbiota in C. elegans, which involves a very conserved pathway. But actually, these sort of mechanisms of regulation of the gut microbiota have not yet been described in a mammalian context. So to recap, we found that the C. elegans quinurenine pathway mediates host-gut bacterium interaction two ways. And this applies not only to contexts of infection, but also commensalism. Some of the quinurenine pathway metabolites are bacteriostatic, others are prebiotic, and this is context dependent. It depends on the microbe we're looking at. And this may actually account for the role of lysosomal organelles in C. elegans immunity. Another implication is that the KP role in host microbe interaction may actually be one of the ways it contributes to the longevity of KNU1 mutant versus KMO1 mutant. So I'm not showing lifespan data, but we also have that. And at the moment, what we're doing is that we are now doing dual transcriptomics on worms exposed to different microbes. And we are investigating what kind of secretory pathway is involved in the release of the lysosome-related organelle content into the worm lumen. And we're investigating modulation of the host quinurenine pathway by bacterial vitamin metabolism. And with this, I would like to thank my team. And a lot of this work actually was carried out by Jack, who has graduated last summer. And now Alejandra has been working on some of these aspects, but mostly on a completely different thing, which is a massive screen, which hopefully will be able to share relatively soon, maybe at the next worm meeting. And I think that's it, that's me done. Should I stop sharing?
Yes. Thank you so much, Dr. Benedetto. Amazing lecture, amazing talk, really. Well, I have a curiosity. Like, I don't have expertise in the microbiota using C. elegans. So I was wondering if you ever checked these metabolites from the quinurenine pathway have any impact in the neurons or neurodegeneration or any psychiatry disease? Because I mean, we know that this microbiota in the gut can be correlated to the brain. So have you ever got?
So I have not done that yet myself. So I mentioned that I started doing some whole brain imaging in C. elegans, essentially, and that will be one of the things I will be looking at in more details. But the Ashrafi lab, as I mentioned very early, has published some of this data. Obviously, it's part of the gut-brain axis, the quinurenine pathway, in mammals as well as in C. elegans. The evidence in C. elegans is just emerging. But obviously it's going to progress really, really fast because worms are just so much faster. And we know that some compounds are very neurotoxic, and actually it's a big focus in mammalian studies and human studies. You know, the quinurenine pathway actually is studied mostly for its impact on the brain. and also to some extent in the gut. But actually, in the gut is quite different from C. elegans because the arm of the immunity that is the most studied is not innate immunity, it's actually acquired immunity in this case. So the carrying pathway has an impact on the sort of second line of defense that the host will develop. And for instance, involved in chronic inflammation with, specific macrophages, monocytes, and all these immune cells that are being differentiated and maintained upon release of specific carrying pathway metabolites.
Okay, so we have another question. So Diana says, thank you for the great talk. Interesting mechanism of resistance. The fluorescence bacteria looks so beautiful. So she asks, in general, is there a difference in the resistant mechanism between the gram-positive and gram-negative? So what about LPS isolated from gram-negative? Do you think that the response would be the same or the bacteria needs to be in trigo?
So that's a good question. So from looking at, for instance, at our bacterial growth curves, we mostly tested gram-negative bacteria so far. But the few gram-positive bacteria that we have put in, basically we can't classify bacteria in response to the carrying pathway in vitro based on their gram staining. So it doesn't cluster this way. So some bacteria, we tested about 50 different bacteria, and they have quite different profiles based on other aspects of bacterial metabolism that have little to do with the fact that they're gram-negative or gram-positive. So the carrying pathway doesn't seem to specifically, at the moment, we don't have a specific pattern that we can just really associate with gram-positive versus gram-negative.
Okay. So we have one more question. So Priscilla is asking, have you attempted to induce gut damage prior to infection or infect C. elegans cells to observe different effects?
So, so our paradigm is we look at we start in young adulthood. We try to have very controlled conditions for starting infection so that, let's say, in different mutant backgrounds, for instance, it's not fully established, but it's kind of known in the community, in the C. elegans community, that bacterial seeding in the gut is highly dependent on the ability of bacteria to pass the pharynx. The pharynx is a bacterial grinder, essentially, and in order to be able to grind bacteria efficiently, the pharynx needs to be active, but also the gaps between the teeth that the worm has in the pharynx needs to be tight enough that they can crush bacteria. What happens is that the worm ages and grows, little gaps are formed that allow live bacteria to pass. So for all our experiments, we have to think about that, and that means we have to take animals that we assume are big enough to be unable to discriminate mechanically between different microbes. So essentially, we work on animals that are more than 24 hours past the L4 stage, and we always start our infections this way. Those animals are always very healthy. I have not yet done infections on old worms to look at, the dynamics there by light microscopy. However, I have done infections on worms on different ages, which was one of the little data reported in the paper in Aging Cell in 2019. And what I found is that there is a peak resistance for infection for C. elegans, which is not exactly the same as the peak resistance for heat stress resistance or for oxidative stress resistance. But yeah, I think it will be interesting to look at infection in, let's say, old age, because that's a primary concern in the case of, I would say, mammalian and human diseases.
Okay. We have another question from Diana. The blood death is a very interesting method, and many researchers still do not know this methodology. Can you talk a bit more about it, like if it works for another stresses?
So that's a good question. So essentially what Diana is referring to is like the kind of like a high throughput assay with a plate reader, and then you record blue fluorescence and you can figure out when your worm population has passed the median time of death. And that can be used to do all sorts of survival assays. The limitation of the assay are actually quite well described in the paper. But so far you can do a number of anything that will kill the worms to 100% of the worm one way or another can potentially be used, unless somehow if it's a compound that quenches the fluorescence or that dissolves the worms, for those compounds it will not necessarily work, because let's say if you dissolve the worms, then the blue fluorescence will not be released particularly in a predictable manner, essentially. So there are limitations there. So some solvents, for instance, we can't do that assay very successfully with them. But it works for most infections, I would say. It works for oxidative stress. It works for heat stress. You have to scale the worm population that you're using with the length of the assay. So basically, the longer the assay, the more worms you need. So the slower the death, the more worms you need. And that is because to detect a peak in population blue fluorescence, you need to have individual worm peak to aggregate. So if the worms are dying one after the other in a very discrete manner, then all what you would get is little blips, and you won't be able to identify a growing peak that will tell you when the median lifespan of the population has passed. Yeah.
Sorry to ask that, but how many worms do you need for each assay?
So, for an assay that would kill in like 6 to 8 hours, 16 worms are enough.
Okay.
For the infection assays, we tend to go toward 100 to 150 worms.
That's a lot. Okay, so I don't think we have more questions for now. So thank you so much, Dr. Benedetto, for your presence here. We really appreciate it.
Thank you very much for the invitation.
Sure. Bye.
Bye-bye.
So once again, I wanted to thank you, Dr. Benedetto, for such interesting talk. I hope you guys have enjoyed it. So I also like to say that if you have any questions, you can go through the chat and you're gonna translate the questions. And right now I wanted to introduce our next speaker for this evening. It's our last speaker, Dr. Chantal Fradin from France. So she is a lecturer at the University of Lille in France and studies the effect of dietary and endogenous advanced glycation end products on health and more specifically on aging. Her research is carried out in Professor Eric Bolligan's team, headed by Professor Philippe Amon. And with a background in parasitology, she has a long-studied host pathogen interactions, work on 2 fungus pathogens, Candida albicans and Coccidoide posadaceae, during her thesis in France and during her postdoctoral fellowship in Medical College of Ohio in the United States, and also in the Robert Koch Institute in Berlin, Germany. Her work is focused on characterizing the molecules that enable pathogens to interact with the host cells and the mechanisms that regulated the expression of virulence factors such as proteins, glycoproteins, and glycans. Since shifting the focus of her research to the biology of aging, she has been working mainly with the C. elegans model. She uses it to highlight the mechanisms that can accelerate aging, such as carbonyl stress, which promotes the glycation of the endogenous molecules, mainly proteins and DNA, leading to appearance of mainly hallmarks of aging, and also ingestion and intestinal absorption of dietary advanced glycation and the products, inducing systemic inflammatory signs. So with that, I would like to welcome Dr. Chantal. We are delighted to have you here today. So thank you so much for accepting our invitation.
Thank you very much for this nice introduction. I would like first to thank Priscilla and Camilla for their kind invitation to present a part of my work to this symposium. I didn't know this symposium, but I think it's a really nice idea, and we should do things like this in France. And then if we have some symposiums on C. elegans already, but not like that. Okay, I will talk about part of the work we are doing on the glycation, and our main study is to characterize the pathophysiological mechanisms of glycation and to try to find targets, preventive and therapeutic targets. And I will use the part I am doing with on the model C. elegans. As we have heard from the first talk, improvement of health and reduction in the frequency of infectious diseases have increased the life expectancy at birth in almost all countries. But there is an increase in the frequency of chronic pathologies with age. And these chronic pathologies can be like type 2 diabetes or kidney failure. And these chronic diseases are associated with low-grade inflammation, which is an inappropriate, uncontrolled, and sustained chronic inflammation, which can be induced by different factors such as aging and unbalanced diet, like the Western diet. And this inflammation involved various inflammatory molecules, such as receptors of the innate immune system. And these receptors can be activated by different molecules, such as pathogen-associated molecular patterns, the PAMPs, and also damage-associated molecular patterns, the DAMs. Oh, sorry, I think I did something wrong. Sorry about this. And I will put back the subtitle. I try to put back the subtitles, but yes, it's okay. And the DAMs include the advanced glycation end products, which are molecules which are produced after the linkage of a carbonyl group from reducing sugar, such as glucose, to a free amine of a protein. And this reaction is a non-enzymatic modification. And these AGEs, then advanced glycation end products, can be formed during aging and also pathological conditions, like during diabetes, with when the amount of sugar is very high, and also when there is oxidation, which can induce carbonyl stress with the production of different decarbonyl compounds, such as glyoxal or methylglyoxal, which will glycate proteins. And this reaction with carbonyl stress is faster than the glycation reaction from the glucose. And then this product, the AGEs, can interact with different scavenger receptors at the surface of our cells. And this AGE could be a source of low-grade inflammation because the binding of this AGE to the different scavenger receptor can induce and maintain inflammation and oxidation. And we know that inflammation and oxidation can also enhance the expression of scavenger receptors and induce the production of AGEs and the secretion of other DAMs, like HMGB1 and protein S100, which are also ligands of these scavenger receptors. And then the inflammation can be maintained. The modification of protein by the glycation reaction can also induce dysfunction, aggregation, and cross-linking inside the same protein or between proteins. And this modification of protein can also induce inflammation and oxidation. It's why in the group we believe, and also other researchers believe, that AGEs are a source of this low-grade inflammation, and this low-grade inflammation associated to aging is called inflammaging. As I told you, an unbalanced diet can also be a source of, is one of the factors of this low-grade inflammation. And we know that unbalanced diet can also be a direct source of AGEs because it's the glycation has been discovered by Louis-Camille Maillard during food processing and during the cooking of the food, and especially food where you have lots of carbohydrates, of course, lipids, and of course proteins. You have this glycation which will take part and which will modify proteins. And then we are eating these AGEs. And some studies have shown that these dietary AGEs could be also a source of inflammation. And we know that unbalanced diet can also induce carbonyl stress, which will then induce endogenous AGEs, as I told you before. Then this unbalanced diet can be a huge source of inflammation. But at the moment, we don't know if dietary AGEs have the same role than endogenous AGEs for activation of inflammatory signals. And it's why one of the hypotheses is that the consumption of dietary AGEs, as well as formation of AGEs when we eat, for example, Western diet, can induce inflammatory signals, which are pathophysiological mechanisms of chronic diseases. But of course, it's an hypothesis, and we need to confirm this. And it's what we try to do in the lab. And one part of my job in the lab is to try to understand how dietary AGEs can induce these inflammatory signals, and also to try to find out how the endogenous AGEs are induced when we are eating this unbalanced diet. And for this, then first, the main goals of the project is then to assess the intestinal absorption of dietary AGEs, because of course, for inflammatory, for induction of inflammation, AGEs need to go through the intestinal barrier, and you will see that it was not clear yet if dietary AGEs can go through or not this intestinal barrier. And also the second part is to analyze the endogenous glycation induced by a high glucose diet, like it's the source that we can find in an unbalanced diet. And for this, we need, of course, a simple model than C. elegans. We didn't choose C. elegans because of the inflammation, because we know that we cannot study inflammation in C. elegans, but at least we can study oxidation, which are usually induced at the same time than inflammation. But C. elegans is quite interesting because first, it's a good model for aging research, and at the end, we are interested to know the link between glycation and aging. But also because C. elegans has a complete digestive tract with enterocytes, and we know that enterocytes are the cells which can absorb the nutrients. C. elegans, in physiological condition, has almost no AGEs. There is no glycation because it grows at a temperature between 20 and 25 degrees. Then at this temperature, the glycation reaction is very, very, very slow. But of course, we can induce glycation in C. elegans in oxidative condition. And also when the worm is incubated in a glucose-rich diet, then it's quite interesting because we have almost no basal glycation and AGEs when with the worms, and it's very good because then we can really see even a small induction of glycation. And to also analyze the dietary AGEs which can be ingested by the organism, it's quite good to have an organism with almost no basal AGEs. And C. elegans has glycation defense mechanisms which are conserved, which are the same than the ones in the mammals, or almost the same. And it also expresses some of the scavenger receptors which are present in mammals. And it's quite interesting. It's then an interesting model to study glycation. Today, I will just present you our work on the intestinal absorption of dietary AGEs. And what we knew when we started this study is that only 3 AGEs could go through the intestinal barrier. 3 AGEs are just glycated amino acid. And this, the mechanisms which allow the absorption of 3 AGEs by the intestinal barrier is the passive diffusion of these 3 AGEs and the glycated amino acid, or also the transport of D or tripeptide, glycated D or tripeptide, by the transporter PEPT1. And after, inside the enterocytes, the D and tripeptides are digested, and only the 3 AGEs is released in the lamina propria. But with these mechanisms, it means that no peptide or protein AGEs can go through this intestinal barrier. And we know that only peptide or protein bound IgEs can bind and activate the scavenger receptors, like the receptor Rh, for example. And we know that 3 AGEs cannot activate the scavenger receptor. Then, if only 3 AGEs can go through the intestinal barrier, then the hypothesis of the activation of inflammation by these dietary AGEs is then wrong. But we wanted to be sure that it's the case because in some reviews, some authors speculate that maybe there is other mechanism of absorption of intestinal absorptions of AGEs, for example, transcytosis or parasitosis, which could transport, or at least transport peptide or protein-bound IgEs. But it was just speculation. There was no confirmation of these mechanisms. Then, for our study, we of course study one AGE, carboxymethyllysine, then it's a lysine which has been modified by sugar, and after several reactions, it gave carboxymethyllysine. And this is an AGE, and we are working a lot on these AGEs, like lots of different scientists, and we know that CML, then carboxymethyllysine, can induce inflammatory signals. And we know also that this CML can be produced both in the body, like it's an endogenous AGE, and it's also produced during food processing. Then it's why it's quite an interesting AGE to study. Then we modified the food of the worm, and we modified two types of proteins. First, the dietary proteins, the normal dietary proteins of the worm. Then it's proteins from bacteria, because bacteria is the main food of C. elegans. And we also modified a protein supplement, bovine serum albumin. It's a protein which has been used a lot in a marine model when people were checking the effect of dietary AGEs on the health of the mice. Then it's why we use also this protein supplement. And to modify these proteins, we incubate them with glyoxylic acid, and we know that glyoxylic acid modifies lysine into a carboxymethyllysine. And we use 2 concentrations of glyoxylic acid to have proteins with different degrees of glycation. And then we, of course, confirm that the bacteria and also the BSA, I don't show this on this slide, but BSA was also, we also confirmed that it was glycated. And here, with an antibody against CML, we could check that bacteria were glycated. And then we fed the worms with this food. And the work I will present you the data obtained mainly with glycated bacteria. And of course, we fed the worms with glycated bacteria and also with the control bacteria. And we did the same with BSA, glycated BSA and control BSA. And after several days of incubation, we analyze different parameters. First, we check by Western blot with the antibody against CML. We check if the worms ingested the proteins. And the answer is yes. We couldn't find CML epitopes inside the worm's lysate, and it was quite nice. And when we remove the glycated bacteria from the diet and we analyze the worms 2 or 3 days later, we could see that there was a degradation of these proteins. Then the worm were eating and degrading these proteins. We know some people were arguing that glycated proteins were less digested than normal non-glycated proteins in the gut, but most of these studies were done in vitro, and in vivo, it was not that clear that glycated proteins were less digested. But anyway, we just check if worms incubated with glycated bacteria have expressed more or less genes coding from digestive proteins. Then we check genes coding for luminal proteases and also lysosomal proteases. And we were surprised to observe that worms incubated with glycated bacteria express more lysosomal proteases. And then we checked if other lysosomal genes were also induced when worms were incubated with glycated bacteria. And we could observe that some genes coding for lysosomal membrane proteins were also more expressed when worms were incubated with these glycated proteins. And we then thought that, as we know that lysosomes are in the cytoplasm inside the cells, we thought that maybe the glycated proteins went inside the cells of the worms. We then set up the immunochemistry in the lab, and with the antibody against CML, we check where were located the CML epitopes. And for this immunochemistry, we check also, then the CML epitopes are in green, and in red, we check a protein which is located in the apical cortex of the enterocytes, meaning that in red, we can see more or less the lumen, then the apical part of the enterocytes, and it means we can see the lumen of the intestine. And what is interesting, and we were quite surprised because it has never been described before, we could see vacuoles inside the enterocytes. You can see here with DAPI the huge nuclei of the enterocytes, as the intestine of C. elegans contain 20 enterocytes, and it's quite huge cells compared to other cells of the worms. And we could see this vacuole around this nuclei, meaning that these vacuoles are inside the enterocytes. And we were wondering if this vacuole could be endosomes. And then we did a colocalization of the CML epitopes with different endocytosis markers. And we could observe that we had a colocalization of the CML epitopes with this endocytosis marker, confirming that CML, dietary CML, seems to have been endocytosed. Then we were wondering if this endocytosis was specific of the glycated proteins. And for this, we did immunochemistry using a lectin, the concanavalin A, which like all lectin, recognize carbohydrates, and Nè can recognize carbohydrates, of course, of C. elegans, but also of bacteria, as you can see here in the immunofluorescence we perform on our control bacteria and the glycated bacteria. And Nè could, we detect endosomes only in worms which were fed with the glycated bacteria. With control worms, the Nè didn't stain any endosomes, meaning that this endocytosis seemed to be specific of AGEs. And we know that, as I told you, C. elegans has some genes which are orthologs of the genes, mammal genes, which code for scavenger receptor, which can recognize AGEs. And we could observe that the worm fed with the glycated bacteria expressed more some of the scavenger receptor, notably SCAF5 and 6, compared to the control worms. And I don't show this in this presentation, but we are doing now some experiment with RNA interference, and we could start to see some inhibition of the endocytosis when targeting at least 2 scavengers, meaning that this scavenger receptor seems to be involved in the endocytosis of dietary CML. And then we just check the worm after feeding them with these different glycated bacteria. And we could see that the worms which were fed with glycated bacteria had lower eggs counts than the control worms. And when we fed the worms with protein supplements, the BSA, the glycated BSA and non-glycated BSA, and in this condition, there was no impact of glycation on fertility, meaning that it was more protein specific than glycation specific. And we were wondering if the protein supplement was also endocytosed by C. elegans, by the enterocyte of C. elegans, and immunochemistry results show us that yes, the BSA CML, glycated BSA, is also endocytosed by C. elegans. And when we check the longevity lifespan of C. elegans in the with glycated dietary proteins or glycated protein supplements, we could see that with the glycated dietary proteins, worms live longer than worms incubated with control bacteria. But it's quite, it's not surprising because as there is less fertility, we know that when there is less fertility, longevity is higher, but the worm didn't look in a very good shape, but they live longer. When incubating with protein supplements, we didn't see so much difference between the glycated condition and the control condition. There is just for the highest, the worms incubated with protein which has the highest percentage of glycation, we have a slight reduction of worm lifespan, but it was a very small reduction. Then we can see that there is some difference, and it's quite intriguing and interesting as well. And then we check other markers like autophagy, and we could see that glycated diet, either dietary proteins or protein supplements, we didn't see a strong regulation of the gene expression coding for this type of markers. And we were also quite happy to don't see so much difference because we were afraid of caloric restriction, and it seems that what we see with glycated bacteria, it might not be due to caloric restriction. And we check also the response, the stress response to the accumulation of misfolded proteins. And we could see that this, the genes coding for molecules of this response were induced when worms were incubated with glycated proteins. And we know that this misfolded proteins response, the misfolded proteins can be due to oxidative stress, and we know that, okay, ages can induce oxidation, and we check the antioxidant response, and notably expression of SOT3 and GST4, which are usually overexpressed when there is oxidative stress, and we could observe an induction of the expression of these genes when worms were incubated with glycated proteins. Then, to sum up, we could observe that dietary CMLs and one dietary AGE can be ingested by worms, and this we were not sure that it could happen, especially for the protein supplements, because lots of people, when I started the work, told me, oh, you know, C. elegans will ingest bacteria and will not ingest other molecules like proteins, other proteins. But yes, it can. And this was quite nice, because we know that this model can be used for our purpose study. We could identify a new intestinal absorption pathway for dietary CML, endocytosis. This endocytosis appears to be receptor dependent, and we are doing new experiments with a fluorescent FITC labeled glycated proteins to confirm that it's really specific of glycation. And this endocytosis seems to depend on a receptor, and we are, as I told you, trying to find out which receptor or receptors are involved in this process. And we could also observe during this experiment, even if it's preliminary experiments, preliminary results, well, the big part of the work was to discover this endocytosis as a new intestinal absorption of AGEs. And we could observe that the glycated dietary proteins modulate stress responses, linked in part to the accumulation of misfolded proteins and also oxidation. Of course, there are several prospects to this study, and we will have to study further the endocytosis of dietary CML, as I told you, to identify the receptors involved. We want also to check what is the fate of endocytic glycated protein. Are they completely digested, and maybe just free AGEs which can really pass through this intestinal barrier? Maybe there is exocytosis and or transcytosis, which can allow the passage of peptide or protein-bound CML or AGE. And this is quite interesting, because in mammals, this, if it's the case, then it means that this peptide or protein-bound AGE could induce inflammatory response. And with some colleagues which are doing lots of analysis, chemical analysis, we will check the amount of free CML versus protein or peptide CML to really check what are the proportion of these different type of CML, and to try to find out if really peptide CML, at least peptide CML, can go through this intestinal barrier. And we are starting work with Bertrand Mérès, who is working in another laboratory in Lille, and he's working with a human enterocyte, and he's also doing a human and murine organoid from intestine. And we will check with him if the endocytosis that we could highlight during our study is a conserved pathway for the intestinal absorption of dietary AGEs. To be honest, I believe it is because we know that the scavenger receptors, notably CD36, and it's the ortholog of the SCAV receptor of C. elegans, CD36 can induce endocytosis of AGEs in other cells, like macrophages and so on. And for example, for the atherosclerose, CD36 can endocyte oxidized LDL, and we know that among oxidized LDL, there is glycated LDL. And what we see with enterocytes looks like what we see with macrophages, and I wouldn't be surprised that it's the case in mammals. And then, of course, as soon as we have identified, really clearly defined the different mechanisms of this endocytosis and the different molecules involved, then we will be able to really check the physiological impact of dietary CML and to try to find out why we have this fertility problem, and maybe to have a better check on the effect of this dietary CML on the lifespans of the worms. And before to finish, I would like to thank all the team from Lille and the team glycation, from inflammation to aging. And the boss of the team is Eric Boulanger. And I would like to thank Constance Dubois here. And she was my PhD student. She graduated last year, and she's working now in Montreal, still on C. elegans. And she has done lots of the work I show you. And she has, as Erika told you, C. elegans is it's not new for me, but I work on C. elegans since 5 years, something like this, and it's quite new. Then with Constance, we learn a lot together, and it was very nice to work with her. And also Charles, who helped also to go further in our study. Thank you very much.
Thank you very much, Dr. Fradin. Amazing talk. We have a few minutes for questions, if that's okay. So Gustavo is asking, great talk. Thanks for sharing your results, Dr. Chantal. Question, autophagy could be a potential mechanism related to the free ages degradation by CML?
Yes, it could be. This could be more for endogenous AGEs, the one which can be which are produced inside the cells, because then when the proteins are glycated, then they might not be really recognized by the proteasome to be degraded and so on, and then they might go through autophagy. Yes.
Yes, okay. And how to control the function of this process in long-term treatment in C. elegans?
The control the function of this process. What does it mean? Because
I don't know. I mean, I don't know. Maybe he's asking like if there is any way to control this process when you treat the C. elegans like for a long-term treatment.
With this glycated diet.
Yes.
Well, the glycated diet is already glycated, and this is quite easy, and we can give glycated diet through all the experiments, or we can just give it for just some days, wash the worms and put them back to a normal diet. And this can be good to really check the degradation of AGEs.
Okay, so one question. From that essay with the lifespan that you showed that increases lifespan, it was a long-term treatment or like acute treatment? Because it was a really interesting result, right? You'd expect to decrease the lifespan.
Exactly. And this was a long-term treatment.
Okay. How many days?
And well, it was all their lives. They were fed with this glycated diet. And we think it's because we could see that the worms, they didn't produce eggs. Well, even if we add a few days that they stop reproducing, but you can still see eggs. And we could see that with the glycated bacteria, we didn't have any eggs, or almost no eggs. And I think it's certainly why the lifespan is...
That's why it's good.
Yes, because there was one paper discussing this and saying when there is lots of fertility, then lifespan is shortened, and the way around.
Yeah. Okay. Another question. Diana says, "Great talk, fantastic work. The endosomes visualization is beautiful and provides great insights. One question. A long time ago, we exposed worms to methylglycoxal and we could not observe any toxicity. Do you think we should have associated with high glucose diet to generate the AGEs?"
Yeah, but this is really interesting because it depends on lots of things, because we did also with methylglyoxal and glyoxal, and that's true that we have to add huge amount of glyoxal and also methylglyoxal. And when we did work with glucose, in some lab, like 10 millimolar of glucose is enough to reduce the lifespan. For us, 10 millimolar, we extended the lifespan. And I think it depends also maybe on some components of our media, like the peptone. Maybe not all the peptones are the same. And that's true that you could, she could have associated high glucose also, because then it would have helped, because it generates also AGEs, even if methylglyoxal is faster than glucose. But as told one of my colleagues, because he's working a lot since a long time on glycation, methylglyoxal acts very fast. Then maybe it will act outside the worm. And then when the worm is ingests methylglyoxal, it's too late, because the methylglyoxal has already reacted with other components in the medium and so on.
Okay, so one curious question about, you showed in the beginning of your presentation that the AGEs must be bind to a protein in order to activate the receptor.
Yes.
Is that right?
Yes.
Do you know it can be like any protein, like any amino acid? Because I was thinking that must be like a specific conformation to activate, in order to activate the receptor. So do you know like any proteins, like if it is specific or no?
And I think it's not so specific of proteins, it's more the length of the peptide, because it can be also only peptide. But you are right also, the conformation is certainly very important, because it's most of the scavenger receptor, like the RAGE, which has been very well studied with AGEs, RAGE can bind different ligands. Lots of ligands. And we could see, because we have a cellular test, and sometimes, I mean, for some tests, we can hardly activate this receptor. Then it's certainly very important, maybe some conformation, and actually we don't know yet, because there are lots of studies, but people don't really know exactly what's going on. And this is a bit of the problem. And also the antibody against the receptor are not very specific. And I think some studies sometimes, okay, talk about rage, but maybe it was not rage. Then it's, yes, it's a good point. And it's why glycation is quite difficult because there are lots of different ages and lots of different proteins and so on. There are still lots to know.
Yeah, well, this is a fantastic work, Dr. Chantal. So thank you so much for being here today. So yeah, I'm going to end the event right now. So thank you very much, everyone, for your audience and the participation today. I would like to thank our speakers today for their amazing lecture. And on behalf of the SIMPECE organization, I'd like to express our gratitude. And right now, I'm going to end the event, so I'm going to switch to Portuguese. So, pessoal, muito obrigada pela audiência de vocês e pela participação. Gostaria de agradecer novamente a vocês, aos nossos palestrantes e ao nosso patrocinador Pixels, que é quem está promovendo esses brindes. Então, a gente estava fazendo sorteio no WhatsApp, quem ainda não tiver na lista, entra lá. Então, a Pixel é uma empresa que ela está utilizando a visão computacional e a inteligência artificial para automação e detecção de padrões na área de saúde e do meio ambiente. E essa empresa está oferecendo diversos produtos para utilização com nematóide, né, com C. elegans, no dia 23. E aí teremos uma breve apresentação dessa empresa, da Pixel, que vai falar um pouco sobre os produtos voltados para o C. elegans, especialmente para análise de comportamentos, parâmetros fisiológicos e controle remoto de temperatura em bodies. Então, vai ser uma parte muito legal. Então, amanhã, não percam. Muito obrigado ao nosso patrocinador Pixels. Também gostaria de lembrar vocês que o link do formulário e a palavra-chave estão nos chats do YouTube, então não se esqueçam de marcar presença, porque é necessário no mínimo de 75% para poder receber o certificado. E amanhã teremos o nosso terceiro e último dia de evento, então amanhã às 18h30, no horário de Brasília, nós iremos iniciar a nossa palestra com o Dr. Mamos, falando sobre toxicidade de metais no sistema nervoso, usando o modelo do C. elegans, e também teremos as apresentações dos trabalhos científicos nos formatos dos pitches. Então, amanhã nós temos muitas coisas, muita programação. Então, mais uma vez, muito obrigada pela participação de vocês e uma ótima noite.
Então, né, eu entrei aqui, né, só para dar uma boa noite aí para todo mundo, né, cumprimentar todos e todas, no lugar da Priscila, né, que está em aula hoje. Então, espero que estejam aproveitando bastante o nosso evento, que quem está participando dos minicursos, né, então esteja aproveitando bastante também, né, e aí acho que nos encontramos amanhã, em especial, né, durante a palestra e os pitches, né, Érica, e também aproveitar para parabenizar aqui a Érica pela mediação, né, que mediar em inglês, em English não é fácil, né, então foi fantástico, viu, Érica? Então, acho que é isso. Uma boa noite, né, para todo mundo, né, Érica? E nos vemos amanhã.
Tá, joia. Muito obrigada, professora. Tchau, tchau. Obrigada pela presença,
Tchau.
pessoal. Até amanhã.